Hi, welcome everyone. I'm Meredith Fensom, Oxitec's head of public affairs and a Florida native. We appreciate you all joining us. We have many registrants for tonight. Most are from outside the area. Oxitec has led community engagements in the Florida Keys for about a decade, and tonight's webinar was advertised in local papers, shared on the radio, and promoted on our social media channels. It was also publicized on the FKMCD and Oxitec websites. The purpose of our webinar this evening is to provide members of the public, especially residents of the Florida Keys, an opportunity to receive information and ask questions about the proposed Florida Keys Mosquito Control District, we'll call them FKMCD, an Oxitec project. As far as our agenda, we're doing the introduction now. We'll have a presentation for about 20 minutes, a panel introduction for another 20 minutes, and then we'll have a Q&A for more than 2 hours and close at 8 p.m. Eastern. So tonight for the Q&A, you should see a Q&A bar on your screen. Please enter your question there and include your name, organization, if any, and your location. Remember to press send and we can answer the questions submitted there. We've received a number of questions over email in just the last 10 or 15 minutes. If during the webinar you could please just use this Q&A bar to submit your questions. It'll help us keep them all organized. And again, we'll continue answering questions until 8 p.m. Eastern if they last for that long. We may group similar questions together to allow us to answer as many as possible. If we get to the end and we haven't answered all the questions, we can then respond by email. We will prioritize questions submitted by Florida Keys residents. Our panelists tonight from the FKMCD are Ms. Andrea Leal, the Executive Director, and Dr. Larry Rebar, the Director of Research. From Oxitec, we have CEO Gray Franson, Head of Field Operations, Dr. Kevin Gorman, and Head of Regulatory Affairs, Dr. Nathan Rose. Our independent panelists include Dr. Oscar Aline, the Chief of Programs and Services for the National Association of County and City Health Officials, NACHO. We have Mr. Bob Eady, the Administrator and Health Officer for the Monroe County Department of Health. We have Doug Mader, a veterinary specialist with the Marathon Veterinary Hospital, and Dr. Jorge Ray, Director and Professor of the University of Florida, IFAS, Florida Medical Entomology Laboratory in Vero Beach. I'll now hand it over to Dr. Kevin Gorman for our presentation.
Many thanks, Meredith. Appreciate the introduction. So the objective of this short presentation is really just to set the scene for everybody, to give everybody the sort of pertinent background information, both on why the proposed project has been proposed, exactly what the time frame is, what the content is, what the scope of the project, to give everybody an overview of the regulatory and the approval process. And I guess most importantly, really, it's to stimulate questions, to maybe feed some questions back into the Q&A session that will follow later on. So why now? We know Aedes aegypti is a significant problem. It's been increasing over recent decades. The Zika outbreak of 2015 that stretched into 2017 was a real eye-opener for everybody. It damaged economies right around the world and certainly had a significant effect on the local economy in Florida too. Dengue has been an ongoing problem for decades, and it's certainly not going away. You can see by the chart on the right-hand side that dengue cases locally in Florida have generally been on the rise over the last decade or so. And even this year, we've got a significant number of cases that have been locally acquired. And then, of course, there are other diseases from Aedes aegypti too. Chikungunya is just one of those, and they can be crippling and certainly give lifelong impact. Environmental impact is certainly a consideration. Every tool has its place. Every tool is very useful in its own way. But making sure that vector control has a minimal impact on the environment, particularly in delicate places like Florida and the Florida Keys, is really very important. Our technology, Oxitec's technology, is very species specific. It only kills Aedes aegypti itself, and it targets females of those of that Aedes aegypti. And we've had very good reviews in terms of its environmental profile, with 9 national and state agencies now concluding that there's no environmental health risk or human health risk posed by the release of these mosquitoes. So the purpose of the project, the overarching purpose is really just to broaden the toolbox for vector control authorities. That's the long-term goal, the longer-term goal. This is a small project. This isn't a project that's being proposed to tackle disease incidents, in any particular locality. This is a project that's been proposed to demonstrate the effectiveness of a tool so that vector control authorities can choose whether to use it at a later date. It's about making sure that those options are there for vector control authorities and broadening that toolbox to make sure that there's a suite of tools that can be used for different situations. Preserving the quality of life for residents, of course, is part of that, and evaluating this innovative tool is actually the way that toolbox will be broadened. It'll lead to regulatory applications if all goes successfully, and then that will enable vector control districts to progress. I see somebody's moving slides. I'll just go back. There are 4 project components. Primarily, it'll start with community engagement, working with local communities to discuss the actual aspects of the trial, the technical aspects. Once they start, there'll be a marked release for capture, which primarily looks at the biology of the mosquitoes, how far they fly, how long they live. And then there'll be 2 small projects where we use single release points and area-wide releases to evaluate the performance. There's a small number of boxes in total, and we expect the whole project to be completed within about 28 weeks. This is a snapshot of a project that we've just completed in Brazil. Again, it was from the boxes. The boxes, as you see on the bottom left side there, contain a capsule of eggs. They also contain some diet for the developing larvae. And it's a just add water approach. Water is applied to the box, and shortly afterwards, around about a week to 10 days afterwards, mosquito males start to emerge and release themselves into the environment. This particular technology was tried out in Brazil, as I mentioned, since 2018. We've had 2 years of trials there now. And the latest year trialed the box and showed that we got around about 90% suppression of the population in 14 weeks from the outset of treatment. We had fully self-limiting releases, so there was no persistence long-term. It lasts for a certain number of generations afterwards, a residual activity, we call it. We got significant suppression, as I mentioned, and we had no female released. We got very good public acceptance, and the lack of females was something that the EPA also examined during their regulatory process.
This big marker block.
This slide here, can everybody mute their microphones? Thank you. This slide here shows the approval process. Oxitec's technology has been reviewed by all the US federal agencies relevant to agriculture and public health. So the USDA approving agricultural insects of ours. Well, the mosquito started life with the FDA back in back in around about 10 years ago. And in 2016, we had a finding of no significant impact for our first generation mosquito. Then this year, we've had the EPA approval for 5034, which is the mosquito we're talking about today, the second generation. And that was followed by the state approval shortly afterwards. The EPA has done a very comprehensive review. We submitted personally about 4,000 pages to the EPA, and they concluded that there was no risk, environmental risk or human health risk from the release of the mosquitoes. They're certainly very excited to see the prospect of a new tool being developed, and they examined all that data as mentioned, as well as all the latest scientific literature, and also getting expert advice externally from institutions and organizations such as the CDC. The state approval, which followed, again, was a comprehensive review. It entailed 7 agencies in addition to the Department of Agriculture and Consumer Services themselves. Those agencies are listed there. You can see that those bureaus and agencies include the Department of Health, Fish and Wildlife Conservation Commission, and the Department of Environmental Protection, amongst others. One thing that we're certainly very pleased about is the independent review of this project. So the CDC have confirmed that they would like to offer us technical support to us and the jurisdictions to review the project, to review the data, and ensure that everything's robust and evaluated correctly. That's a really great asset to the project, and we're super excited about the CDC's involvement. In addition to that, we've got an independent advisory board, and that consists of the University of Florida representatives, the Department of Health from the state, and also Doug Mader from the Marathon Veterinary Hospital, an established peer in the community. So a very nice blend, we think, of independent advisory board members, as well as the overarching review of the CDC to supplement FKMCD's review of the project and the evaluation. This is a snapshot of the timeline, if you like, the window that the project fits into. So you can see the approval process on the left-hand side, and 21st of July in the middle there is the date of the FKMCD Commissioners' vote. So next week, next Tuesday, there'll be a board meeting where those commissioners will vote on whether to approve the investigational agreement for the project. If that is successful and it does get approved, there's the potential that the project could fit into the window between that vote, essentially the end of 2020, all the way through to 2022, till about April 2022. And so what will we be doing? Well, the project will consist, as I mentioned, of a single point and multi-point releases. We'll replicate those. We'll have them compared to untreated areas. These are all protocols reviewed and assessed by the EPA and developed in conjunction with them. The locations of those projects will be determined, once we look at the actual monitoring of the surveillance of the pest pressures there, and we'll also obviously talk to the communities too. In terms of what we're going to evaluate, it'll be dispersal and longevity for some of the biosafety elements relating to the mark release recapture. We'll be looking at what's called residual activity, the duration of effect once we've stopped releases of mosquitoes. And we'll also be looking at the presence of these mosquitoes in natural breeding sites to see if they actually pervade the environment and access those areas that are difficult for normal conventional chemistries to be applied at. We'll be assessing the percentage kill of the female mosquitoes. So of the treated mosquitoes, what's the percentage of mosquitoes that die, of female mosquitoes that die? And also the percentage of the population treated. So of the total population there, what proportion is treated by our male mosquitoes and what proportion remains untreated? This is a schematic of what it might look like on the ground. On the left-hand side, you see the smaller mark release recapture. It's a single point release. And on the right-hand side, you see the larger multiple point release. They both actually cover a similar kind of area if you think about the dispersal of the mosquitoes and the trapping systems that we'll have to monitor them. The single point release is a single box which will be replaced every week. We'll have replication up to 9 small areas, and we'll monitor the progress of those mosquitoes, both using adult traps and ovi traps or egg traps. On the right-hand side for Project B, there'll be multiple point releases. There'll be maybe 10 to 15 boxes in each of the areas, depending upon the requirements and the pest pressure there. And they'll each release mosquitoes every week, and we'll replace them on a weekly basis. It's about 12 weeks for Project A, and it's about 16 weeks for Project B. There is the potential to overlap them, but in running sequence, they would take us around about a minimum of 28 weeks. So we should be able to complete the whole lot within one single mosquito season, providing everything goes well. As I mentioned before, in terms of the locations, they will be selected depending on pest pressures. We'll look at use the surveillance to look at those pest populations and ensure we understand what those levels are. And then we'll also talk to the different communities involved and make some decisions following initial community engagement. These things can take a little bit of time, of course, and we've been working now with the FKMCD for 10 years. It's been a great relationship for us. It's been very transparent, a very strong footing for a project to start on. And that certainly will continue. We will be hand in hand as we go through the community engagement programs. We use a whole range of tools, and we'll be tailoring those to the local communities, directed and led by FKMCD, but with the experience and input of Oxitec, who've been doing these kind of community engagement programs for many years. We'll be trying to involve the community as well as just informing and listening. We'll be making sure that the whole group of stakeholders are engaged, informed, and remain so right throughout the project and after the project too. We'll ensure that there are ample opportunities for people to contact us as well as for us to contact them. And this last slide really just recaps on the referendum that was in 2016. You can see here the 33 precincts lined up in a bar chart, and 31 of them voted in favour. It was a total of 23,422 votes in favour out of a total of 40,000, just over 40,000 votes. So a pretty strong confirmation that there was a need for a project, there was a desire for a project, and in the two districts that voted against, they were still close calls, as you can see. And so we'll remember this as we go through and start to talk to communities and decide upon locations in conjunction with the surveillance data as we go forward, providing that the vote is successful next week. And with that, the presentation concludes, and I will hand back to you, Meredith, to move on to the next agenda item.
Thank you, Dr. Gorman. We'll now introduce our panelists. And for the FKMCD and Oxitec panelists, Ms. Leal, if you could please start. Okay. We may have lost her. We will go on to Dr. Rebar.
April.
Dr. Rebar, I'm not sure we can hear you. Could you just briefly introduce yourself, your position with FKMCD? Okay, I'm not sure that Dr. Rebar can hear us. Mr. Franson, could you please introduce yourself? and Dr. Gorman.
You're not having much luck, Meredith, I'm afraid.
No, I'm really not. I mean, luckily we can all see the names, faces, and positions on the slide. And I did briefly introduce them earlier, but you all in the audience will be hearing from these panelists tonight. So I wanted to give them a brief opportunity to introduce themselves.
Well, we can certainly come back and give them an opportunity as they reconnect. My name's Kevin Gorman. I've been with Oxitec now since 2012. I run field operations, which means essentially all the insects that are out in the field come under my accountability. I'm a public health entomologist as well as an agricultural entomologist by training, about 20-something years in the public sector before Oxitec. Nathan.
Hi, my name is Nathan Rose. I have a PhD in chemical biology from the University of Oxford in the UK. At Oxitec, I look after the regulatory affairs portfolio. So in the context of this project, I've helped to steer the EUP applications through the EPA and the Florida state processes. And I've been with Oxitec about 4 years now, and my background is in molecular biology, particularly that focus.
Thank you very much. We'll move along.
Meredith, I can see Gray's reconnected.
Yeah, can you hear me, Meredith?
We can.
All right, nothing more to say than just hello, and I'm Gray, and I'm Oxitec's CEO, and have been for about 2.5 years. So thank you.
Thank you. Okay, we'll move on to our independent panelists, and we'll ask them to speak for a few minutes to introduce their position and role, and also their interest in this project. Dr. Aline, could you begin, please? I cannot hear you.
I think you just have to speak really close to the microphone, Oscar.
Okay, let's move on to Mr. Eady. We'll come back to Dr. Aline.
All yours, Bob.
I'm not hearing Mr. Eady. Are others hearing?
No.
Okay.
No.
Okay, we'll come back. Dr. Mader.
I'm here. Can you hear me all right?
Yes, thank you.
All right. Excellent. I'm Dr. Doug Mader. I'm a veterinary specialist at the Marathon Veterinary Hospital in the Florida Keys. I'm a true conch. I was born in the Keys many years ago. Moved all around the country, came back here in '94, and have lived here ever since. I love this place. I love the environment. We're an incredibly lucky community to live in such a beautiful place. And taking care of this environment is extremely important to me. It's one of the reasons I get up in the morning. For those that don't know me, I'm actually a triple boarded veterinary specialist. I'm boarded in dogs and cats. I'm boarded in zoo animals, and I'm also boarded in a specialty of reptiles and amphibians. So I have kind of an eclectic interest. I'm an adjunct faculty at 2 veterinary schools, written 4 textbooks, published over 40 peer-reviewed papers, 2 or 3 dozen book chapters. Point being is that I'm not just giving vaccines and spaying puppies and kittens. I have an academic background. I have a master's degree, and I understand science protocol and peer-reviewed journals, and I'm objective when I look at data. I'm on this panel because I'm taking a totally different viewpoint than everybody else. I'm a veterinarian, and something that you don't talk about, you don't hear much about. I mean, you hear about dengue and Zika and chikungunya, and these are all seriously horrible diseases. Look at the news, the media from the last few weeks. Look at the surge in dengue cases. What people don't talk about is the vector diseases that we see in dogs and cats. Heartworm disease is an incredibly huge killer of dogs and cats, and we have a heavy, heavy presence of it here in the Florida Keys, and it's carried by mosquitoes. Well, we've had like 11 cases of dengue in the last few weeks. We average about 1 case of heartworm disease in dogs every week in our clinic. That's 50 cases a year. And not only dogs get it, but cats can get it, ferrets can get it, other mammals can get it too, most in the canid family, which we don't have too many here, foxes, coyotes. But believe it or not, and most people don't know this, but humans can get heartworm disease as well, carried by the same mosquito. So I look at this as a huge issue for me as a veterinarian here in the Keys. My goal when I wake up in the morning is to protect that human-pet bond. And that means anything I can do to keep that bond alive, I'm going to go ahead and do it. And if there's a way to prevent a disease which is potentially lethal, such as heartworm disease, I want to get involved in this project and see what I can do to help. So that's why I'm here. And I appreciate the opportunity, Meredith and the panel, for letting me be with you.
Thank you, Dr. Mader. We're happy to have you with us. And Dr. Ray, over to you. Dr. Ray, I'm not hearing you. Are others hearing Dr. Ray?
No.
Let's come back in a minute. Dr. Aline, can we try you now? I can't hear you. I'm sorry. Okay, what we'll do, let's go ahead and move to the Q&A. And I'm sorry that we haven't been able to introduce all of our panelists, or that they haven't been able to introduce themselves, but hopefully we can figure out something with the audio. They weren't on mute, so we should have been able to hear them. But we will move on to the Q&A, and I'm looking for questions. We received a number of questions by email before this began. Sorry, give me 1 second. And I may turn to a couple of those first, and then I think that you're grouping some of these questions together for us to be able to respond to. So I'll be on the lookout for that. But in the meantime, we received a number of questions from Danielle Hill. She's a resident of Key Largo. And the first question, which perhaps is best answered by FKMCD and Oxitec in conjunction, is about the current means by which FKMCD uses to control mosquitoes. It's her understanding that this will continue during the project. And she asks, how will Oxitec and FKMCD assess that this experiment is working if the same control is used now is used during the experiment? Doctors Gorman and Rose are prepared to respond to this question, but if our panelists from the FKMCD, Ms. Leal or Dr. Rebar would like to speak up as well, please let us know. I think there's space for both of us here.
Meredith, I'm not seeing Dr. Rebar or Dr. Leal. I'm happy to take the question.
That sounds great. Thank you.
Great. So yeah, it's the same wherever we go and we do a project. Actually, we don't actually impose that the standard control methods that are being applied or may need to be applied are changed in any way. And it will be the same here. We have untreated and treated areas. and the idea would be to treat them both the same if there are additional treatments that are required, because, for example, there are cases of disease which need to be treated. If that's the case, then we would be recording all treatments. It's part of the protocols. It's part of the stipulation by the regulators that all treatment is recorded. Those would be submitted to the regulators, and that would be taken into account during the interpretation of the results.
Thank you, Dr. Gorman. Okay, we have additional questions, and this next question was asked by Danielle Hill, who is in Key Largo, and it was also asked by Megan Hull, who I believe is in the Keys. She's a Florida Keys resident, and they have both asked how Oxitec will manage unintended consequences if there are any. So we'll turn this to Oxitec. Dr. Rose, could you respond to this question, please?
Sure. Are you hearing me okay?
Yes.
Okay. Thanks for this question. So what I'd like to do is just go back to the way that regulators actually assess what sort of consequences might be expected from these releases. In their assessment of this particular project, the EPA looked at potential consequences for human health, potential consequences for animal health, and potential consequences in the environment as a result of releasing these mosquitoes. And this was a very thorough analysis which they carried out. So they looked at the impact of releasing male mosquitoes and how these male mosquitoes might interact with humans. They looked at any potential toxicity or allergenicity that might be associated with the mosquitoes and found that there was none and that there was no risk to human health as a result of these male mosquitoes being released. They also looked at a wide range of environmental impacts that might be expected from these mosquitoes being released. So obviously the most important one is what happens if you reduce the mosquito population as might be expected. And their assessment here comes back to the fact that these mosquitoes are invasive species in Florida Keys. They've only been in this environment for the last few hundred years. So by reducing the population of this particular species, you're actually restoring the environment to what it would have been like before these mosquitoes invaded Florida. They come originally from Central Africa. They looked at particular impacts on a wide range of animals that mosquitoes might interact with or that might eat mosquitoes. They looked at a wide range of impacts on animals if you were to remove these mosquitoes from the environment. So in all of these, the EPA and then subsequently the Florida state authorities carried out a very, very comprehensive review. And their conclusions after this review was that there was no risk to human health, to animal health, or to the environment. And as I've said, they've looked at a very wide range of potential impacts of these male mosquitoes being released. So coming back to the question, what about unintended consequences? The regulators here have studied this very extensively. The other thing to say is that this is not the first time that these mosquitoes are being released. This particular strain of mosquito has been released for 2 years in Brazil, and it's been very successful at suppressing wild mosquito populations. After wild mosquito populations are suppressed, after the project stops, the wild mosquito population eventually will recover unless those releases are continued. And there have been no negative impacts on human health or on the environment as a result of the mosquito releases in Brazil. The main outcome of those projects was successful suppression of the wild mosquito population as expected. Kevin, I don't know if you have anything you'd like to add to that.
Just that I'd say that, over 1 billion Oxitec mosquitoes released into the environment now, multiple countries over 10 years, and with no adverse effects noted at all. So I think, you summed it up really nicely, Nathan. But the long history of releases and the number that was released gives us, should give everybody assurance that there are no likely unintended consequences.
Thank you both. And we received that same question also from Deborah Wilson. We're not sure where Deborah is located. But another question that we have received is also from Danielle Hill in Key Largo, is whether there is going to be any type of genetic monitoring program to help detect any unanticipated outcomes or consequences. So similar to the question before, but this one asked specifically about genetic monitoring. And she notes, especially since this new version is supposed to be female killing only. So we'll send this question to Oxitec colleagues. Dr. Gorman, could you start for us, please?
Yeah, absolutely. Genetic monitoring is something that we're working towards, I guess. We're talking to universities at the moment, specific universities at the moment, about putting a genetic monitoring program in place. Yet to be confirmed, Meredith, but we're keen to get something off the ground. So that would likely be taking samples before, during, and after the releases to look at the genetic makeup of those populations and to see if there's any change or introgression. This is a second-generation mosquito. We expect that all the males will survive, or at least the vast majority of the males will have the potential to breed and integrate background genes. That's part of one of the advantages of the technology. We have insecticide susceptibility in our strain, and we'd like to see that pushed into a resistant population so that we can make conventional chemistries more useful again. There's certainly a good amount of insecticide resistance in the local populations in Florida, and it would be great to be able to show that we can have a positive impact on that.
Could I just add something there as well?
Absolutely, Nathan.
Yeah, so the other point here, so the regulators have not requested a genetic monitoring program. They have requested other types of monitoring. So making sure that we are tracking the mosquitoes in the wild, that we see their disappearance from the wild after the end of the project, as would be expected and as we've seen in Brazil. But the regulators did actually assess the potential for the introgression of background genetics into the wild population as part of their assessment. So EPA looked at this, and they also sought input from the CDC on this particular question. And after assessing this and concluding that it is likely that there will be introgression of background genes into the wild population, their conclusion was that this does not add any risk to the project. So the mosquitoes which will be released, as Kevin said, are susceptible to insecticides. They also don't have any added ability to transmit diseases in their background genetics as part of that particular mosquito strain. And so on. So this is something which has been specifically addressed by the regulators and again concluded no risk to the environment or to human health.
Thank you both. And are any of the other panelists interested in adding anything? Perhaps Dr. Rebar, just if you want to. I just want to give the opportunity before we move on to another question. Okay. We'll go to the next question.
Meredith,
yes.
Before you go, this is Bob Eady. Can you hear me now?
Yes.
Okay, good.
Could you please introduce yourself, Mr. Eady, now that we can hear you, and just briefly talk about your position and role and interest in this project? And we'll do the same with Dr. Elaine if we can get him on after you.
Okay. I'm Bob Eady. I'm the Administrator and Health Officer for the Florida Department of Health in Monroe County. And as the health officer for the Keys, my interest in this is, as everybody will remember, we had the first locally outbreak of locally acquired dengue back in 2009 and 10. Fortunately, that went away, but sporadically we get dengue again. And this time we have locally acquired dengue appearing in the Upper Keys in the Key Largo area. And that is despite the efforts of the Mosquito Control Commission and the Health Department. this does happen. But my interest in this is that there is no one way to successfully stop an outbreak, unless, which is impossible, unless you can figure out a way to eliminate all breeding sites for the mosquito in that area. And given that a bottle cap full of water is enough for the mosquito itself to breed in, it's almost impossible to do. So in the interest of the environment, also from my perspective, is what other tools in a toolbox is the best way to look at it that I've always seen do we have, and I have been interested since the beginning of Oxitec's efforts in the Keys as to see how it was going. I am very interested in what goes on now. I'm interested to see how it goes. I am very optimistic that this project goes forward and then it will prove successful, because there is no way that you can spray yourself out of a situation, because if you do, then you're impacting the environment in such an adverse way, and there's just really no other ways to. We're losing the tools that we have regarding chemicals to be able to control mosquitoes like this. So I'm really interested in this. I need to point out that I am not in any way connected with Oxitec, nor am I endorsing Oxitec or anything. I'm looking at this procedure and this technique to evaluate how it will work in the Florida Keys. But I will say that I have indeed for the last 10 years been talking with my colleagues in public health. They are supportive of this sort of technology and the development of it. And, if we go forward, then, or if science goes forward, we need to be willing to see what kind of reasonable experiments we need to take to protect the public health. As we see in this country right now, for years, it has been warned that we could have a pandemic such as the COVID outbreak, and we have one now. It would be nice if we could have a vaccine, but we don't. I think it's also interesting that at least for 2 decades, there has been an intensive effort to develop a vaccine for dengue, and that's been unsuccessful. So we need to figure out how we can eliminate the mosquito that carries the disease and transmits it from one human to another. So I'm very optimistic about this project, and I hope that it goes forward and that it comes out very successfully. I haven't been able to find any downsides that concern me in any way. Most of the questions that have been raised have been dealt with by expert in various regulatory departments. The Department of Health, I have been in contact with, and as you pointed out, they're supportive of the project. So I am not advocating for Oxitec, but I am advocating for any way that we can control mosquitoes.
Thank you, Mr. Eady. We appreciate your interest in the project. And Dr. Aline, if we could do your introduction now.
Oh, no.
No, we can't hear you. I'm sorry. I thought we had this figured out. You're not on mute, but we can't hear you.
Can you hear me now?
Yes.
Yes.
Someone muted me. Okay. Okay. So, good evening, everyone, and I'm sorry for the technological delays from previous My name is Dr. Oscar Allen. I'm the Chief Program Officer for the National Association of County and City Health Officials, called NACHO, not NACHO for short. And we represent the several 3,000 county and city health departments, just like our colleague Bob Eady represents here in your jurisdiction. Specifically, I have been, as I can say, an environmentalist at heart and an epidemiologist by passion. I spent 16 years in New York at the beginning, middle, and towards the end of the West Nile virus outbreak in 1999, all the way up to 2015, I'm sorry, with respect to working as the Director of Epidemiology in the New York County Health Department there, and specifically working to help develop both a comprehensive mosquito control program and a vector control investment strategy that totally relied upon community engagement, as well as strengthening the local department's capacity. Since that period of time, I can say the lessons we learned, both from West Nile and chikungunya and dengue and all of the others, I have now become one of the chief leaders at the NACHO, responsible for addressing several programmatic support across the country. A portion of that has been extensive work around Zika. And we were able to, when at the height of the Zika outbreak, identify that over 90% of the communities who had vector control programs were not necessarily in a state of readiness, if we can use that term, for Zika. And that level of concern is very high of how we can help support jurisdictions to not only increase their state of readiness, but utilize the tools that are going to be important for them to be successful, especially if they're not able to invest significant capital into the addressing of vector control issues. So like Bob has stated, I have there's no connection to Oxitec in a sense as far as not advocating or giving any formal endorsement, but specifically the role of being here as an independent SME is also to observe and to participate, because what happens within your jurisdiction, we imagine will be able to be either replicated or even used as a best practice across the continental U.S., especially for those low resource communities who are desperately looking for a way to improve and really strengthen their vector control program, while really focusing on what are those strategies that can be low on the chemical inputs and high on the overall health outcomes, so that we can protect the health and safety of the communities where people live, work, and play. Thank you very much.
Thank you, Dr. Allen of NACHO. We appreciate your perspectives. Thank you. And Ms. Leal, can we try to introduce you now?
Yeah, can you hear me?
Yes, can hear you well, loud and clear.
All right, so I'm Andrea Leal. I'm the Executive Director here at the Florida Keys Mosquito Control District. I've been with the district for 17 years and have been the director since 2016. Throughout that time, I've kind of gone through the ranks as far as what I've done here at the district, but went through the dengue outbreaks that we saw in 09 and 10, and have really spent a lot of time and energy trying to figure out, the best way to control these Aedes aegypti. I think it's important to mention, why we look at these new technologies. And I say new technologies kind of in a lump because, we are looking at many different things. Like many others have said, this is not a smoking gun. There's not one thing that's going to help, finally control these. It's really going to be a combination of tools. Currently, we use quite a few things, but unfortunately, we're seeing resistance in our local populations. So it's really pushing us to kind of think outside of the box and evaluate some of these new technologies that are coming available to us. We've done trials previously with Lobachia, had great results, and at this point in time, we're really just awaiting product registration to move any further with that. I know we're in constant contact with that company, and we're already working on a plan, hopefully in the near future, once product registration occurs with that. Following very closely the sterile insect technique that's going on to our neighbors to the north in Lee County. So really just wanted to emphasize we are looking at a number of different things and feel that these tools hopefully will give us, what we need to supplement what we're already doing to help control Aedes aegypti.
Thank you, Ms. Leal. And Dr. Ray, could we try to come back to you?
Can you hear me now?
Yes, perfect. Thank you. Loud and clear.
Yes, I changed all the microphones. Anyways, my name is Jorge Ray. I'm the director of the Florida Medical Entomology Laboratory. I have a PhD in biological sciences from Florida State University. Our center, the FMEL as it's known, it's a research and education center for the University of Florida. We're located in Vero Beach, Florida, and it was established in 1956. We conduct basic and applied research on arthropods that transmit diseases, such as mainly mosquitoes, ticks, sandflies, and on occasion others. Our research includes basic and applied research on all aspects of this topic, including ecology, biochemistry, molecular genetics, modeling, and many others of both vectors and the diseases that they carry, which basically allows us to explore a lot of interactions between vectors, diseases, the environment, that other units that only concentrate on one aspect of the problem look at. The center has a staff of 11 full-time faculty members at the entomology and nematology department of the University of Florida. We have a number of graduate students, undergraduate interns, research technicians, and support personnel. We work closely with mosquito abatement agencies, public health agencies, and environmental groups and agencies. And I think my decisions to participate in this project, I think other people have mentioned before. It's another tool in an arsenal that is diminishing, and I mean an arsenal for the control of disease-carrying arthropods. The number of chemicals that are available is going down. Some are disappearing from the market. Others are becoming ineffective because of our resistance. We have several projects on our lab looking at resistance in Florida and elsewhere, and it is a very serious problem. And the development of new products, and by that I mean chemicals for control, it's either slow or non-existent. So this is, as Andrea mentioned, just another tool that may be very handy in the very near future. That's it.
Thank you, Dr. Ray, for the introduction and for the perspectives. We appreciate it. We appreciate your interest in the project. Dr. Rebar, could we return to you for a brief introduction?
Yeah, hello, Lawrence Rebar, currently Director of Research here at the district. I've been here 22 years. Prior to that, did a number of things 30 years ago, actually 29 years ago. I actually was at the Florida Medical Entomology Lab where Dr. Ray is. Mrs. Leal, Mr. Eady, and Dr. Ray, all three pretty much said the things I wanted to say, but some of them bear repeating, I guess. My personal concern is insecticide resistance by Aedes aegypti. In the past 30 or so years, we have seen, I mean, worldwide, just a proliferation of populations that are resistant to what few chemical pesticides we have. To run a truly integrated control program, you've got to test and, if proven useful, employ every control method that's available because it's been mentioned by several other people. You know, the toolbox, as we call it, it's shrinking. Every year there's a question of whether products are going to be registered, if there's anything new available. We've really got to look at anything that comes along and evaluate it, if it's something that we can use or not. Thank you very much.
Thank you very much, Dr. Rebar. I think everyone's been introduced now. Have I missed anyone? Okay. Well, we've got a lot of questions, so we'll return to those. And again, we are prioritizing questions from Florida Keys residents. We've gotten questions from people outside of the area, including about malaria, and we're not going to respond to those now. But the next question says, this is from several people. We've grouped this question together. It's from Barry Ray, who's from the Florida Keys, Megan Hull, who is also a Florida Keys resident, Danielle Hill in Key Largo, and they have said the scientific reports, I believe they're referring to the article, in scientific reports suggest that hybrid mosquitoes will be more robust and better at spreading diseases. Is this the case? We can turn to Dr. Gorman at Oxitec for this question, unless any of the independent panelists may want to respond. Okay, Dr. Gorman?
I can take this one, yeah, but I think Nathan's probably got it covered better, so I'll pass to Nathan,
Okay.
scientific reports one.
Okay, so just to give a bit of context for those who are listening to the webinar and aren't familiar with this, a paper came out in September 2019 from some scientists at Yale, together with some scientists in Brazil, that claimed that some of Oxitec's mosquitoes in Brazil had made the wild mosquito population more fit and better at transmitting diseases. This paper was later discredited. The editors of the particular journal that published it published an editorial expression of concern in March this year, where they re-reviewed some of the conclusions that the authors of that paper had made. It turned out that on the re-review, the authors had actually substantially gone beyond the data that they had generated in the paper and speculated way beyond what the data were actually able to support. So just to answer that question directly, there was no evidence in this paper or from any other studies that have been done that Oxitec mosquitoes, when they mate with wild ones, will make wild mosquitoes more vigorous or more fit. In fact, the opposite is true. This paper actually showed that the wild mosquito population after Oxitec mosquitoes had been released was suppressed as expected, but also the remaining mosquitoes were actually less fit than other wild mosquitoes that were present in Brazil. There was also no increase in the ability of the wild mosquito population to transmit diseases. There was no increase in insecticide resistance, and in fact, the paper addressed both of these directly. They actually showed that there was no ability to transmit diseases more effectively, and they referenced papers that had showed that there was actually no potential for the increase in insecticide resistance either. So to answer that question directly, there is no concern of increased hybrid vigor or fitness of the wild population, and there is certainly not any risk of increasing insecticide resistance or the ability to transmit disease. This is also something which the EPA deliberately and very carefully assessed when they assessed this particular mosquito, which we plan to release in Florida. And they conducted that assessment together with the CDC and came to the same conclusions. Again, no added risk to human health or to the environment on any of these particular measures.
Thank you, Dr. Rose. Dr. Rebar, or would you like to add anything here, or would anyone?
This is Lawrence Rebar. Just a little bit, not really. A lot of times there is an assumption that what people call hybrid vigor is a given, and it's not. I mean, hybrid vigor, really, that's a term from crop breeding where, they're trying to develop plants that produce better yields or, have a perhaps a better resistance to some plant pathogen. But they're, that's those are things that are produced in a controlled setting. They're deliberately people are deliberately manipulating the cultivar for an agricultural purpose, and these things a lot of times they survive better on a farm, but they may not survive well in the wild. There's also a phenomenon that you can sometimes see in some populations where actually the hybrids are actually less successful than parent strains. There was a study about 15 years or so, and it was using a largemouth bass, but it turned out the hybrid strains are actually more susceptible to infectious disease than were either the parental strains. And going back to this particular publication in question, they really didn't present any evidence that was the case. I believe they worded it that it may. Well, a lot of things may. That doesn't mean they are. Thank you very much.
Thank you, Dr. Rebar. Would any other of the panelists like to say anything here? It's okay if you don't. We have lots more questions.
This is Jorge Ray. Well, just one small point, and that is that determining the ability of a population of mosquitoes or any vector to transmit diseases is an extremely complicated process. It's not something that you do as a sideline on another process. I mean, it may take years because there's a huge number of factors involved. So the fact that somebody's saying they're better at transmitting disease, it's not very credible from an experiment of this type.
Thank you, Dr. Ray. We'll move on to our next question, unless anybody else would like to speak. Okay, we'll move on to our next question. Before we do, for all of the audience watching, just want to make sure you know that there's a little scroll bar on the bottom. So all of the panelists who are on video, you can scroll left to right, and you should be able to see them speaking, even if they're not appearing on your screen or seem to as you hear their voices. There should be a way for you all to scroll at the bottom. So just wanted to draw attention to that. The next question, and again, we are prioritizing questions from Florida Keys residents. This is from Jessica Hogan. She is a resident of Key Largo, and she asks, where is Oxitec intending to conduct the releases? The Keys are a large area, and the public should be informed of the general area of release locations. It's possible we'll have a couple of responses to this one. Before I turn it over to Oxitec's head of field operations, Dr. Kevin Gorman, are there any other panelists, perhaps from FKMCD, who might like to say something? Okay.
Sure, Meredith. I can jump in if that's okay.
That's great.
Yeah, so we've been doing a lot of mosquito collections for the last several years at this point. But we will make sure that the public is informed as to where this will be occurring once any decision has been made. At this point in time, no decision has been made. We're really focusing on areas where we're seeing enough population of Aedes aegypti, as well as looking at that 2016 voter referendum. It kind of showed us where folks were more inclined to accept this technology. So we will just make sure that when decisions are made, everyone is informed. We will be going door to door in those areas, making sure we're contacting residents, and again, just being as transparent as we possibly can be.
Thank you, Ms. Leal. And Dr. Gorman?
I think Andrea said it all, really. I think it's all about pest pressures, making sure that we've got the mosquitoes there to evaluate ours against. But also, obviously, doing that initial community engagement and making sure that we have the support and those areas will be selected in conjunction with FKMCD, primarily by them.
Thank you, Dr. Gorman. We'll move on to our next question. It's somewhat related. Says, is there a risk to humans from exposure, and don't we need consent from exposed individuals? And two of the people asking this question, one is Barry Ray, who was from the Florida Keys, and the other is Dana or Dana Pearls, who I believe is from the area as well. I think either Drs. Gorman or Rose could respond to this. Are there any independent panelists who would like to say anything on this point? And this is about human exposure and consent.
Meredith, I'm happy to take that one if no one else wants to.
Great. No, that sounds good. Go ahead.
Yeah, so this is a question which also came up in some of the public comments on the application. So the EPA addressed this in their response to public comments. So I'll come to their response in a moment. But the first point to make is that this is not an experiment on humans. Oxitec would be releasing male mosquitoes only. And as I'm sure many of the people listening to this know, male mosquitoes do not bite. They don't actually have the mouth parts to bite. Only females can bite, and only females can transmit disease. And this particular strain of mosquitoes is designed such that only male mosquitoes are released. So they do not bite humans, and therefore they don't actually carry any risk to human health. That's the major point to make here. EPA answered the question about whether informed consent is required. And EPA's finding was that this particular research project involved with the release of these mosquitoes does not meet the regulatory definition of research which involves human subjects under the applicable regulatory standards. And so EPA's response to this question of informed consent was that this doesn't meet that standard, and therefore this is not required. And that matches up with the fact that these mosquitoes are actually not interacting. They're not biting humans.
Okay. Thank you, Dr. Rose. Anybody else? Yes.
Just not that I would contradict the EPA, but I am on the Department of Health for Florida's Institutional Review Board, and I would concur with that question. This is not such an activity that would be covered as research on human beings. So I don't see that there is an issue, and it would not be subject to having to file a request for it to be reviewed by the IRB. But I think any question along that line would have been resolved by the EPA when they did their review themselves.
And just to add also, that's really helpful. Thank you, Mr. Edie. The same questions would have applied to the release of Wolbachia mosquitoes, which again are releasing male mosquitoes only. And again, EPA did not find that required informed consent. So this is a very similar situation to those trials which have already been carried out in Florida and in other parts of the country as well.
Thank you, Mr. Edie and Dr. Rose. This next question will likely go to Oxitec. It's a multi-part question, and it's been received from a number of people in the Keys. And these are questions about the differences between the first and second generation mosquitoes. And okay, multi-part. One question is production issues in Cayman. They're asking if there were production issues in Cayman. Females released. Increased vigor we've already talked about, so we don't need to revisit that now. And then how has the second generation mosquito addressed these issues? So any production issues in Cayman and release of females. Dr. Gorman, would you or Dr. Rose want to start with this one?
And I can take this one.
Okay.
Yeah, super. Many thanks for the question, and I'll just pull up that slide and hopefully you can see that. So we've had a long history in the Cayman Islands, a long and productive history in the Cayman Islands. We've been working there for a long time. We've had 4 projects on the Cayman Islands, and the first one in 2009 was actually the first ever GM mosquito release worldwide. It was a huge first back in 11 years ago now. And it was closely followed by the first GM mosquito performance trial, where it was actually looking at the possibility of it suppressing a population. 2 major firsts, two big wins, if you like, for science, and yet relatively small projects. Since that time, there's been 2 further projects, one in 2016 to 2017, which was a small area-wide deployment looking at pest suppression. And then finally, back in 2018, there was also a project looking at pest suppression, but in this case, it was in the context of integrated vector management. That particular one was the first time that GM mosquitoes had been combined actively in a project to look at a combination approach, integrated vector management. A number of research firsts, and very important, they have been very productive projects without a doubt, and it's been a very productive collaboration with the MRC over there, the Mosquito Research and Control Unit. In terms of production problems that were, to tackle that one first, in the 2016 project, which gave us a 62% suppression value, which is in itself is a good number, and probably exceeds what most chemicals could achieve, given the same situations, a single hit, if you like, a single season. That was also at the same time evaluating a brand new mobile production unit. So a brand new mobile laboratory built in the UK, shipped out to the Cayman, installed on the island, on the MRCU unit there, next to the unit there, and then used for the first time to run that project. So there were some teething troubles with that production unit, and the project, overall was a great success. We got good suppression, although it wasn't the best number, but it certainly was a steep learning curve on our mobile production units, and the potential for deploying those more widely. So the second project in 2018 used the same unit, but at that point, all of those teething troubles had been ironed through, and that was a very successful project again. Full compliance, no issues with, every batch of insects is checked prior to release, the number of females assessed, and that's documented and reported to the regulators, and there was full compliance as usual. So production issues are an inherent issue with all of technologies that are biological. When you're looking at live insects, especially if you're having to produce them, turn them into adults and release them as adults, there's a lot of husbandry involved, and there is a learning curve there. We've done a lot more than most companies, but it's something that a lot of companies struggle with when it comes to biological control. And the performance in the Cayman Islands, was very good on the whole, as I say. So very productive. And so hopefully that's covered females released as well. We have every single batch checked, and we will with the 5034 too, despite the fact that this is a male-only release system. We will have a full quality control on each batch of eggs. That's production issues and females released. Were there any? Did I miss a third part to the question, Meredith?
No, I think that's everything. Well, the increased vigor we already talked about. So no, I think we've got that question covered.
Yeah, one thing I would add is that, in addition to our mutual collaboration with MRCU that's been very productive, MRCU just the other day, Jim McNally, the director there, issued us with this statement to pass on to everybody today, just emphasizing really that positive relationship and the fact that recent projects have been successful. They've progressed through the steering committee, they've reported everything to the regulators, they've had full compliance. And as that active stewardship committee gets through the data and gets to an agreement on how to actually publish those, they will no doubt come out in due course.
Thank you. And we've got another, and it's really a follow-up to that one, is were female releases a mistake?
No, I'll take that one. Female releases for 513A for the first generation, and bearing in mind, please, that all the Cayman projects were using 513A. None of them have used 5034, the second generation. All the projects pre-2018 in Brazil used 513A. Panama used 513A as well. And there's an element of female release in all of those because 513 has incomplete penetrance. So there's also manual sorting as a result. So we had a sorting threshold that was reported to regulators. It's a very tight threshold, and every batch is checked to make sure it's compliant. But there is a tolerance there, and those female releases are reported in all the publications. So they're not just reported to the regulators, they're reported in the publications too. We have lists of publications, and I think I've got one here. I'll just find the right slide. So they are field publications there, those ones in the table. And you can see the 2 Cayman projects, 2009, 2010, followed by a Malaysia project, then 3 in Brazil, and then 1 in Panama. And female releases, where relevant, are reported in all of those.
Thank you, Dr. Gorman. And if whoever is managing the slides could keep that slide up for a minute, because it answers part of another question we received about Oxitec's plans for peer review ahead of release of 5034. Is there anything more you'd like to add other than what's on this slide about independent third-party scientists and peer-reviewed studies?
Nathan, go ahead.
Yeah, just so I think that's a question which came from a number of people as well. The first point I'd make is that Oxitec's technology across all of the insects uses the same 2 genes. So there's a self-limiting gene and there's a gene which makes the insects fluorescent. And Oxitec's technology has been subject of more than 100 peer-reviewed studies. So studying these 2 particular genes and how they work in insects, whether it's mosquitoes or whether it's a range of other insects. So that answers a lot of questions when it comes to anything to do with safety and risk assessment, and also efficacy of these genes. Specifically with 5034, we supplied, as Kevin said, a significant amount of data to the regulatory authorities, to the EPA and to the Florida state authorities. That was reviewed by the EPA's independent scientists and also by the CDC, and then within Florida by 7 different Florida state departments and agencies. So there has been substantial peer review of the 2nd generation mosquito specifically, in addition to the very extensive peer review which has already been conducted across Oxitec's technology platform. In addition to that, the studies which we have carried out in Brazil, which have really just finished in the last couple of months, those will be the subject of forthcoming peer-reviewed papers as well. So that data will also be published and made available.
Thank you, Dr. Rose. Just want to give any of the other panelists an opportunity in case they'd like to say anything before we move on to the next question. It's okay if not. Okay. So this next question, this question has come from Barry Ray, Megan Hull, and Danielle Hill, who are all in the Keys. Dana Wilson or Dana, not sure where they are located, and others. And this question is about the use of tetracycline and the spread of antibiotic resistance in the Keys. And there are a couple of parts to this question. The first is, will Oxitec use tetracycline in this study? The second is, will Oxitec test for antibiotic resistance in this project? And then, if not, why not? Dr. Rose, I believe I'll hand this question over to you. If any of the other panelists would also like to weigh in, just let us know, and you can.
Okay, thanks a lot for this question. This is something which we've certainly had raised before. So what I want to do first is just talk through how Oxitec's male mosquitoes are made, because this will set the stage for answering some of these questions. This particular strain of mosquito is designed such that only male mosquitoes will be able to live and survive in the absence of a tetracycline antidote. So that means that if we want to produce large numbers of mosquito eggs, we need to have females that can lay those eggs. And so what we do in the United Kingdom in our Oxitec facilities here is we feed the female mosquitoes a small amount of tetracycline when they are larvae. And this is on this slide in the top left-hand corner. That tetracycline at the larval stage allows the female larvae to survive through to pupal stage and then through to adulthood. And once they're adults, those females can then produce the eggs, which we need for this particular project. So the amount of tetracycline that would be used to produce all of the mosquito eggs required for the entire project in Florida would be less than 5 grams of tetracycline. That is all used in the United Kingdom, and that's just to produce adult females in the laboratory setting, and those females are then mated to produce mosquito eggs. Those mosquito eggs are then surface sterilized with a sanitizer that is actually significantly stronger than hospital-grade disinfectant. That's part of the egg conditioning process. Those eggs are then packaged into mini capsules, which are shipped to the deployment location, in this case, Florida. And then those eggs are then placed into these release devices. Water is added, larval diet is added, and that device is placed in the field. So at all of the stages after the top left one, there is no tetracycline usage. So there's no tetracycline when the males and females in the United Kingdom are pupae or when they're adults. The eggs themselves do not come into contact with tetracycline, and there is no tetracycline in the boxes which are put into the field to produce male mosquitoes in Florida. So at no stage will the male mosquitoes which will be released actually have come into contact with tetracycline type antibiotics at all. Okay, so that just gives some context. The EPA and the Florida state authorities, and before them the FDA, looked at this particular question and concluded that there is negligible risk that the use of these mosquitoes in Florida would spread antibiotic-resistant bacteria. They responded to a number of questions raised about this by the public, and just to specifically address this, there were at least 9 mentions of this in their response to public comments, and they responded in detail to all of those mentions which came in from the public. Just to go, if we can jump to another slide later on, just to slide 13. I'd also like to contextualize this just a little bit here, because I think this may be helpful. As I've said, the eggs which were shipped to Florida have never been in contact with tetracycline. The amount used in the United Kingdom is a very small amount for the entire trial. By contrast, in Florida, farmers are authorized to use approximately 88 million times more tetracycline for treating citrus trees and other trees to deal with particular diseases associated with that type of agriculture. So the contrast between what is already used in Florida and what will be used in the course of this trial is pretty stark. Also in the United States every year, tetracycline is extensively prescribed for infections, and often for infections that don't actually need antibiotics. And a large amount of it is also supplied to livestock and pets. So the amount of tetracycline usage within the state vastly outweighs any that is used by Oxitec in a different country. So I think that just helps to provide a little bit of context there as well. Just to go into a little bit more detail about why the EPA reached the conclusions that they did. So they assessed the production process, and they assessed also what happens to the eggs once they're actually put into the box and how those male mosquitoes develop. And the fact that there is no tetracycline present in the box where those male mosquitoes are developing is actually quite important, because to maintain tetracycline resistance in the bacterial population, you need the antibiotic present. Antibiotic resistance in bacteria is something which is quite costly to bacteria generally. It affects their ability to actually multiply. And so if you have bacteria which are resistant to an antibiotic and you remove that antibiotic, they will very quickly lose that resistance. So EPA, in their conclusions, also looked at the fact that there is actually no tetracycline usage in the course of this trial either. I don't know if anyone else, any of the panelists would like to add anything to that. Otherwise, I'll hand that back to Meredith.
You know, I think if I can, I'll just quickly from a generic standpoint. You know, it is an excellent question with respect to the concern we've had around antimicrobial resistance, and I think the comprehensive answer here was, you know, quite remarkable. But I would say, as a community, it's very good to see that, you know, there are questions that are held concerning even the impact of, you know, just the prevalence of antimicrobial resistance organisms in any community. So I would commend the question and, of course, commend the answer as well. Thanks.
Thank you. Just to add as well, I think the follow-up part of that question, which I missed, Meredith, was why does Oxitec not test this? This is something which has been assessed by regulators at multiple levels, and with each of them having concluded that there is no risk associated here, that's the reason why there is no need to actually test further for this either. If regulators require specific data on certain things, then Oxitec will, of course, comply with those requests from regulators.
Okay, thank you very much for that thorough response. Another question that we've gotten from a couple of people that we may have responded to this already, but it's, can you tell me how mosquitoes were able to breed with the wild mosquitoes in Brazil since they were all supposed to die? And this is, I mean, this is referencing, I think, the scientific reports paper.
I can take that one as well.
Yeah, go ahead, Nathan. I'm easy.
Yeah, so just again for clarity, this is referring to the first generation of Oxitec's mosquito, OX513A. The second generation, which is what this project is planned for, will definitely have male mosquitoes surviving. That's the design. With the first generation, Oxitec has always been very clear in its regulatory applications, in its communications, and also in its published peer-reviewed articles, that with that first generation, a small proportion of the offspring would survive, usually 5% or less. And so it was those 5% or less of survivors which were then able to be able to actually continue to breed with the wild population. And so there was no surprise there to Oxitec that was happening. And the findings of that particular paper were also not surprising to Oxitec. And likewise, there was no surprise to the regulators in Brazil either. They were fully aware of that and had taken that into account in, again, their own assessment of that particular mosquito in Brazil. So with regard to the first generation, it was something which was expected. It was something which was shown to happen, and also something which didn't carry any risk for the reasons we discussed earlier with regard to hybrid vigor and so on. With the second generation mosquito here, we fully expect that the male mosquitoes that carry this gene will survive, that the male offspring of those mosquitoes will also survive. And this is obviously part of the risk assessment which has been carried out by EPA. And it's also, we think, one of the benefits of this technology is that those male mosquitoes can actually pass insecticide susceptible genes into the wild population and actually restore the usefulness of some of the chemicals which are used to control this mosquito. And as we heard from some of the other panelists earlier, chemical tools are starting to fail, and the local mosquito populations are resistant in some cases. And so that's actually quite an important benefit of this particular technology as well. And that's only possible because the male mosquitoes survive and can pass on their genes.
Maybe I'd just add, Meredith, that that it is a non-persistent technology, so the transgene doesn't persist, and that paper only documented, quite rightly so, the integration of background genes. So these are just the wild-type genes, essentially, that were detected in the local target population. As the population suppressed very strongly, down by about 90-something%, that level of integration of background genes, of just standard wild-type mosquito genes, is a certain level, and then as the population expands again, as the trial finishes, of course, it's left a certain percentage of the population that looks slightly inflated, but as the paper published, it then died out and disappeared fairly promptly following that. So, exemplifying the fitness costs that were associated with that and not any hybrid vigor.
Thank you, Doctors Rose and Gorman. Would any of the other independent panelists like to add something here? It's okay if not. I just wanted to provide the opportunity. Okay, let's see. We have another question from Jessica Hogan in Key Largo, and she asks, what threshold will Oxitec use to consider the experimental treatment effective? And if deemed so, what are the next steps? I could see us having more than one person respond to this. Let's see, Dr. Gorman or Rose? Okay. I'll hand it on to you well, unless anyone from FKMCD wants to speak.
I think maybe I can start and others can add, because it's a fairly broad question. Yeah. But I think, has the project been successful? The answer is, I guess, is the results are for the regulators, ideally. It's the regulators who will decide whether this kind of technology is something that's made available for vector control districts to use more widely as they see the data come through. So they don't prescribe any specific number. They don't say, okay, 75% your bar here, and if you get 74, it's a failure, and if you get 76, it's a success. They'll look at the data more holistically. They'll look at the single point, the multiple point. They'll look at the range of data and decide whether it's a tool that could be effective or effectively used, maybe in conjunction with other tools. So there's no cut and dry answer, I think, in terms of what the regulators would see as a specific success. In terms of ourselves, we obviously want to see a strong suppression as possible. But the primary goal of these particular trials is to look at some of the other aspects too. It's not just about what percentage of the population we can cover. It's looking at the percentage of effect on individual mosquitoes. It's making sure that that in this environment they're behaving and acting as we would expect them to. So there's a whole gamut of things that we're looking at, and that suppression is not the only thing that will be most successful or not internally as a team.
Can I just add to that as well? The question was, what are the next steps? So as Kevin said, this particular project is aimed at demonstrating the ability of this technology to work in the United States. And that is important because to be able to register a product for use across the country with the EPA, you need to generate data with that product in the United States. And so data from this particular project would form part of a regulatory application to the EPA for a product registration. As Andrea said, some of the other related technologies, if you want to call them that, Wolbachia, for example, are in that process right now of registering with the EPA. After having completed trials in the United States, it's a similar pathway for us.
Andrea, is there anything you want to add, as someone who'll be evaluating this and, with the prospect of actually using it in earnest?
Sure. I think looking at it out of a mosquito control perspective, we look at the same thing for any product that we may use. You know, how effective is it? What is our percent control? And then we have to balance that with ultimate cost. So how many people is it going to take to deploy? What is the product cost itself? And those are answers right now that those are questions right now that we cannot answer. And we can't answer them really about any of these novel technologies that are not registered and for sale yet. So as mosquito control looks at different products, those are the questions that we ask ourselves. You know, what kind of suppression rate are we getting? We added some new tools to our toolbox in 9 and 10 that cost us, you know, about $250,000 a year, but we're seeing, you know, about 50 to 60% suppression of populations by using that tool. So, you know, we do a cost benefit analysis with anything that we're looking at, and we'll definitely measure our use of that by all of those different factors combined.
Thank you all. Any additional comments on this question? Okay. If not, we have a number of other questions. So from Russell Dewey Jacks, who I believe is in the Upper Keys, and he says in the Upper Keys, when the wind is out of the Everglades, mosquitoes blow in and they get really bad. These would be non-GMO mosquitoes replacing those here. How would this be mitigated? So I can turn this to Dr. Gorman, unless any of the other panelists would like to respond.
Okay. Well, I'm taking the question as, you know, assuming it would be in operational use. I guess that's the way I'm understanding it, as if these mosquitoes were being deployed. How would you prevent other mosquitoes coming in and spoiling the ground that you've already cleared, if you like? I guess the answer is that migration of mosquitoes, you know, into areas is something that's pretty much inevitable. As with all pest control, they're typically there for a reason, and when you try and remove them, they typically come back for the same reason, because they're, you know, that's their niche, and they're successful there. So the rate that they come back is really dependent upon case by case. Some areas are more isolated than others. If it's a wide deployment as opposed to a more localized deployment, you might have more chance of having a longer a break or a longer period with only a low presence. With this technology, I would say that one of the good things about it is that the males go out and search for the females. So whether the pest there is at very low densities or very high densities, the males can find the females, and it's a fairly efficient way of applying. And as the population goes down, the pest pressure goes down, you can reduce the number of males that you release, and therefore you can tailor your costs accordingly, and your efficiency is still high. And then, you know, as you succeed and you get to the point where it's essentially a maintenance program, you have then the option of potentially increasing your monitoring so that you detect any resurgence quickly, and you can use the appropriate control agent. That may be a chemical for a hotspot, or it may be continued low-level releases of an Oxitec Mosquito, for example. So there are a number of options available for a sort of monitoring phase approach when the bulk of the pest pressure has come down, and you're really protecting against a resurgent population that's due to migration from outside areas.
Thank you, Dr. Gorman. Anyone from FKMCD like to weigh in?
Yes, Lawrence Freebar. The mosquitoes that the gentleman's referring to in his question are salt marsh mosquitoes. They're not the peridomestic container inhabiting mosquitoes like Aedes aegypti. They have a very different larval habitat. They have different requirements for their other phases of their life cycle. They're not even, they're in fact, they're in completely different subgenera. There's not going to be any competition or replacement of Aedes aegypti by Aedes taeniorhynchus. They simply do not utilize the same larval habitats. That's all.
Thank you, Dr. Rebar. We'll move to the next question if there's not anything else. And the next one is related, and this one may be a good one for FKMC to respond to, at least in part. Maybe Ms. Leal, it says, are there other endemic species of mosquitoes that also transmit these same diseases? How long does suppression of Aedes aegypti population last after releases are completed? Or is continuous release required? This is a multi-part question. Will this GMO project reduce the use of insecticides and larvicides? I'll read through it again just so we have all the parts, and there may be several people responding on this one. But again, it's Cynthia Lewis, Florida Keys resident. Are there other endemic species of mosquitoes that also transmit these same diseases? How long does suppression of Aedes aegypti population last after releases are completed? Or is continuous release required? Will this GMO project reduce the use of insecticides and larvicides? So multi-part question, but all related.
Well, I can kind of start, and then I believe either Dr. Rose or Dr. Gorman can finish. As far as other vectors that are found for the same diseases that we've been discussing, we do have a very small population of the Asian tiger mosquito, Aedes albopictus. It's found in isolated areas in a couple different places in the Keys. We're currently working to get them back out of here. They've not really been established in the Florida Keys. And when we do find them, it really is kind of, throw everything at it to get these out of here. So we're really not concerned with other vectors as far as transmission of dengue, Zika, chikungunya, et cetera goes. And I'll also kind of touch base on whether or not this product would lessen our adulticide and larvicide use. The majority of our adulticide use is in response to salt marsh mosquitoes, which Dr. Rebar just kind of touched upon. Unfortunately, they do migrate in from salt marshes. We see massive migrations from the Everglades in certain wind conditions as well that we would have to still respond to with adulticides. I would anticipate aerial larvicide, which is something that we do specifically for Aedes aegypti, aerial liquid larviciding, that to be reduced. And, obviously we do quite a bit of adulticiding if we have suspected cases or unfortunately confirmed cases of these diseases. We'll respond with adulticide. So really, I think we would see a bit of a decline, but our major adulticide use again is for a different species.
Thank you very much.
Great. Maybe I'll just add and pick up on the how long does it last. It sort of flows from the response to the last question, really, in that everywhere's case by case. We get somewhere in the region of 5 to 10 generations of residual activity with this second generation mosquito. With the 513A, the first one, as soon as we stopped releasing, the males died. All the progeny were dead by the next generation, essentially, and we had almost a drop off the cliff in terms of efficacy. Of course, you would have taken up egg banks and a number of other things, so you did have a sort of protracted effect following releases, but that's certainly extended with the second generation, where we have that multi-generational effect as the males continue through. So 5 to 10 generations of residual activity, and that's one of the things that we'll be looking at during this proposed project, is the amount of residual activity that we get. It's something that the EPA are very keen for us to look at.
Thank you, Dr. Gorman. Would anybody else like to weigh in? I know Dr. Ray has a lot of expertise in this area, but we may have already completely answered the question.
Yeah, I think it's covered well.
Okay, we've got more questions coming. Thank you all. The next one, I think this will probably go to Oxitec and FKMCD. Said when will mosquitoes be released in the Keys? I guess when might these Oxitec mosquitoes be released in the Keys? If we could respond to that.
Yeah, Meredith, I have a timeline slide here, which just might help. So this really just shows the timeline of the project, if you like. Remember, we have this window between approval, obviously subject to a vote by FKMCD board, and the end of April 2022. Extensions are permitted by the EPA, so should an extension be required, it's something we could apply for. But the current project window extends to the end till the end of April 2022. And within that window, depending upon a whole number of factors, operational factors, the season, the mosquito season, as well as obviously the local the global pandemic that's affecting the local environment. Once subject to approval, we'll then go through a planning phase where we plan out these particular steps. But we have at least one full mosquito season in 2021 within that window, and we anticipate, should that be all that we have, we should be able to get everything completed within that one season. Is that okay, Meredith?
Thank you, Dr. Gorman. Yes, I was having trouble getting my mic off mute.
Andrea, is there anything you want to add there?
Yeah, would anybody else like to add anything?
No, Kevin, I think you covered it all. Again, where we are in the regulatory process is we're still waiting for our local board approval. So once that occurs, then we can maybe harden our timelines. We're already in almost the end of July at this point in time. So the amount of time that it's going to take for proper community engagement, setting up the trapping, and doing releases, it's really probably going to push us out of this mosquito season and into next.
Thank you all. The next question is somewhat related, and this comes from 5 or 6 people, including Danielle Hill in Key Largo, Megan Hall, who's also in the Keys, and several people who live outside of the Keys. Says, what data will Oxitec collect, and will it be shared publicly? That question, I think it'll be more than just Oxitec collecting the data, but perhaps Oxitec and others could respond. Dr. Gorman, would you like to go first, or would one of our other panelists like to start?
Andrea, would you like to go first, or I'm happy whichever way?
No, you go ahead and go first.
Okay. So we're collecting, a range of data. I'll just pull up another slide. Hopefully, adjust while we're on there. We're collecting a range of data. The left-hand project there, as I mentioned earlier, is a mark release recapture. It's how long do the mosquitoes live, how far do they fly, and how far do the offspring go? Because of course our males can make females, and then the females can travel a distance and also lay those eggs. So we'll be using egg traps and adult traps to capture or recapture the mosquitoes and assess those biological parameters, as well as the persistence. So once we've stopped releasing, how long are those mosquitoes there for, and what does that decline look like? How long does it take for them to remove themselves from the environment? We know it's roughly a half-life, and we know that it'll be less than 10 generations for the actual presence to subside. In terms of performance data, we'll be looking at whether the female offspring die. So we'll be collecting the offspring from mated females, wild females that lay eggs. We'll assess them for fluorescence, and we'll see that, say, a percentage of the population was treated because they're fluorescent. They were essentially fathered by our male mosquitoes. And then we'll be looking at those individually to see what the percentage of those treated females is that dies. So are we getting 100% of the females dying, or are we not for some reason? And of course, all the data that we have so far has shown that we do. And we'll also be looking at, as I mentioned before, the percentage of the population that we actually treat. So we'll be looking at the percentage of fluorescent insects that are out there, a percentage of the population that's been fathered by our mosquitoes. That'll give us an idea of coverage, essentially whether the application rate is right to get the desired effect. So that's the main bulk of the data that we'll be collecting. There are other bits and pieces of data that we'll be collecting on the side of some of those experiments. In terms of publication of the data, we've obviously got the independent panel who will be reviewing the data. We've got CDC, we've got the regulators, we've got FKMCD, all of which are independent bodies who will have eyes on that data, apart from ourselves at Oxitec. When it comes to publication of the data, then it will obviously go into peer review at some point, providing it's novel enough to actually stimulate the journal, the editor of the journal, and get him to publish it. And we expect that it probably will be, because it's a fairly new, 5034 hasn't had that many publications out, or won't have done by then, because we've got the Brazil trials. And so, peer review will be the way forward for putting it out publicly, but there'll be a whole bunch of independent organizations who have eyes on the data, as well as the regulators themselves.
For the mosquito control district portion, I can assure you all that we are going to be heavily involved in every aspect of this project. We want to make sure that, like Kevin said, we are seeing all the data. We're involved in collecting the data. We're assisting in identifying traps. We're participating in quality assurance. We'll be going through hundreds of traps, identifying all of the mosquitoes that are collected by species. So, we'll know if there are any concerns. I know we've had some questions about albopictus coming in. So we'll be able to monitor for that through all of these traps. Again, everything will be identified, and we will have eyes on it. As we feel that we're independent third parties as well. Obviously, we would like, if we move forward with the trial and our board votes to move forward with the trial, we would love to see the success of the project. But as a district and as a district being taxpayer funded, we need to make sure that we're utilizing things that are working and that we're going to get the most bang for our buck. So, we are going to be very heavily involved in the trial.
Okay, thank you all. I'm looking at the questions. It looks like we may have run out of local questions. We do have questions that are not from locals, but if you are local and if you're putting anything in the Q&A, make sure that you identify where you're from so we can prioritize those. I think that we've answered all of those, and I'm just double checking. We received a few local emails right before this event began, and one of these, if we've already answered this, stop me, but this question is also from Danielle Hill, and she's in Key Largo. It says, is there any data or evidence that this process works to lessen the populations of disease-carrying mosquitoes? And if not, will there be studies with outcomes done during and after this experiment? And she says the previous experiments showed after initial releases, populations were larger than at the start of the experiment.
Meredith, maybe I'll take this one. There is a slide here which we touched on before. It just shows the actual performance in terms of suppression that this particular 5034 device released Mosquito, if you like, using the egg release devices, actually achieved in its last trial, which was through the mosquito season in Brazil last year. And this is a 4-week mean, this line here, and it goes up to about 92% at its peak. So that means that we've clumped together each of the 4 weeks so that it makes it a more robust number. People will be unfortunately familiar with these kind of 4-week means and 7-week means and weekly means that we see in the pandemic statistics at the moment. So this is essentially combining 4 data points to make sure that it takes out any good spikes, if you like, and levels the data and smooths that line out a bit and makes it more difficult to hit a high as well as hit a low. And you can see that we got the start of suppression from very early on, just 3 weeks of treatment. At that point, we don't know that it's going to go the direction that it did. We anticipated in this case from our first generation mosquito that it would take us somewhere in the region of 10 to 12 weeks to really get off the ground. But the very sensitive release, the good fitness of the mosquitoes, and maybe the point in the season that we started the releases, we saw really excellent establishment in the wild population. And as I say, within 14 weeks, we were at 90% in that particular study. That was a great result for us, and we'd love to see the same again in Florida.
Great, thank you. Anybody else wanting to add anything here? And I'm toggling between screens for some of these questions. And I think we have one that could be good for Dr. Ray about albopictus. Well, here, I'll ask it and we'll see who is the best person to respond. It's not from a local, but I think we've about exhausted the local questions. And it's, what would you do if there was a transitory increase in Aedes albopictus during an experimental release? So that might be a multi-part answer by multiple parties, but we are getting some questions about Aedes albopictus. I don't know if Dr. Ray, you might like to speak a little bit about the Aedes albopictus and the Aedes aegypti, and I mean the differences and what you're seeing in the area.
Yeah, and I think Larry and Andrea can also speak to that on that particular area. Initially, when albopictus came into the state of Florida from Texas, it displaced Aedes aegypti from a lot of places, and there are a number of different explanations from plain competition for resources to survival of the larvae in different conditions to satirization of aegypti larvae, but aegypti female by albopictus male. So it's a really complex issue. Now, from what I understand, the interaction between the Oxitec aegypti and the wild type albopictus, it's only going to be confounding depending on the data that you're going to gather, but I don't think that it's going to affect the outcome of the experiment. You can clearly tell the two apart. It's not like they're very similar. You're going to confuse them. And Dr. Gorman, and correct me if I'm wrong, but they're two different species.
Thank you, Dr. Ray. Would FKMCD perhaps like to mention anything here, or we can go to Dr. Gorman?
This is Lawrence Rebar. Don't know that I understand what the question actually is here because, well, it's like Jorge Reyes said, you know, you're dealing with two different species. Now they've got a more similar biology, but in the Aedes albopictus has a very spotty distribution here, and they're usually not around in very large numbers. And again, it would be very difficult to confound data because they're very easily distinguished in the adult stage. They're very easily distinguished in the larval stage, and with a little, and I mean a little bit of practice, not a lot, a little bit of practice, you can tell them apart in the pupal stage. They're and this is all just under a standard, you know, binocular microscope under just, you know, not a lot of magnification. So I don't see that presents any sort of a problem. Thank you very much.
Thank you, Dr. Rebar. Okay.
Could I just add something there as well?
Sure.
Just going back to the question about what might happen if Aedes aegypti populations decrease, would we expect to see any albopictus moving into that particular environment? So in Brazil and in Panama, when we've actually looked at this particular question, we haven't seen any evidence of this happening. So in Panama, that work has been published, and we can share the reference with regard to that. In Brazil, the work has been submitted to the regulatory authorities, but not yet published. But in Brazil, over at least 4 years of reducing the Aedes aegypti population, we didn't see any increase or niche replacement by Aedes albopictus, which is present in Brazil. And as Dr. Rebar was saying, we will definitely be able to look at the presence of albopictus in the field trial sites in Florida to be able to see if that's happening or not.
Thank you, Dr. Rose. And that was Dr. Nathan Rose. I don't think we can see him on video now, but that's Oxitec's head of regulatory affairs. I did want to mention in the Q&A box, we've received a large number of submissions from Barry Ray. They're not all in the form of questions. We're responding to questions. We have gotten another question from a local Keys resident. This is from Deborah Wilson. She's in Marathon, and she asks, will residents be able to opt out of this experiment? Can they refuse to accept a box on their property? And I believe I'll turn this to FKMCD to answer first, if that's okay, or Drs. Gorman and Rose could respond as well. But Ms. Leal,
Sure.
Thank you.
Yeah, so I can handle that, and then either Kevin or Nathan can elaborate. But our goal, should this trial move forward, is to make sure that there is, again, hefty community engagement. We'll be going door to door in areas. We'll be talking to all the residents, you know, and obviously we're hoping that at the end of the day, we've got, you know, similar results to what we saw in the 2016 voter referendum. So, you know, we're going to go to areas where the community hopefully is interested in this. And because these trials are small, we will definitely focus on placement in areas where the public is happy with that. So, you know, we don't anticipate forcing anything on anyone. We want to make sure that the community is happy with what we're doing, not just in a trial like this, but what we do for mosquito control every day. So we definitely want to work with everybody and ensure that everybody is happy at the end of the day.
Thank you.
Nothing to add from me, Meredith. That was a great answer. Great.
Okay. Another question we've gotten, and I'm I'm not sure if this is from a local person, but it's an interesting question, and it may give us a chance to hear from Dr. Mader. But if you don't want to answer it, that's okay, Dr. Mader. We can turn it over to Oxitec. But the question is, can you elaborate on why there is little to no impact on other animal species? And Oxitec is happy to take that one. I just wanted to provide you an opportunity, Dr. Mader, just in case.
I'm not sure what they're asking. I mean, if they're releasing males that don't bite, they're not going to have an impact on any of the species that would be irritated by the mechanical bite. That's the only thing I can guess that they're asking. If the question asker can elaborate more specifically what they want. I mean, some of the issues that we have when we're talking about other species, because when we're dealing with eradication of pests, for instance, when we had the screwworm issue several years ago, they had to be very careful about what kind of insecticides they could put in the Keys that might damage some of the more sensitive species like Miami blue butterfly and the Bartram's hair streak. So, but those are apples and oranges. So what you're talking about dealing with releasing of the male mosquitoes, it really will have no impact on any other species.
Thank you, Dr. Mader. And it was Christina Sperry who asked this question, and I'm trying to keep up with the chat box, but if she does have any elaboration, we're happy to see that. And if any of the other panelists would like to add anything here, that's welcome.
Maybe I could just say that in terms of effects on other species, in the food chain, Aedes aegypti really isn't, a primary food source for any predators. And so, although, it can be a component of certain animals' diet, it's certainly not the staple diet for anything. And it's an invasive species. It really shouldn't be there in the first place. So there are plenty more mosquitoes out there. And as we've talked about, things like Tinniorhynchus are in huge volumes, Culex are in huge volumes in comparison to something like Aedes aegypti or Aedes albopictus in this case. So locally, there's lots of mosquitoes left for things to eat and prey on. Should we reduce the abundance of Aedes aegypti?
Okay, thank you very much. We're getting some other questions in this vein, and if they're repetitive, I apologize. I mean, we won't repeat anything we've already answered, but this is from Harry Apple on Big Pine, Apple or Appel. He's on Big Pine Key, and he's asking, are there any known side effects from these mosquitoes being consumed by the natural predators that prey on them? Birds, reptiles, other insects. It's Harry Appel, Big Pine Key. I don't know if Dr. Mader or others from Oxitec or FKMCD or anywhere, really any of the panelists may want to speak to this. And this question is known side effects from these mosquitoes, the Oxitec mosquitoes, being consumed by the natural predators that prey on them, which could include birds, reptiles, other insects.
Meredith, I'm happy to take this one.
Yes, Dr. Rose, absolutely. I mean, you know this backwards and forwards. And if any of the other panelists would like to speak after, they're welcome to. Thank you.
Yeah, thanks a lot for this question. So this is something, again, that EPA specifically looked at in their risk assessment and something that we provided quite a lot of experimental data to them on. So, for example, we did an experiment where we compared the impact of eating the GM mosquito larvae with non-GM mosquito larvae by fish and also by other aquatic invertebrates which are relevant to mosquito larvae and mosquito pupae. And so we saw no difference in the impact on those fish or those other invertebrates, whether they were eating the GM larvae or whether they were eating the wild mosquito larvae. And this just bears out, again, the fact that the two proteins which have been put into these mosquitoes are non-toxic to any animal that might consume. And so this is definitely something which has been looked at very carefully. The other thing I just want to say is that, as Kevin was saying a few moments ago, Aedes aegypti is not a major part of predator diet as far as we're aware. So, for example, bats will tend to eat larger, more nutritious insects, things like beetles or moths. Birds, quite a lot of studies have been done, and mosquitoes seem to form a very small part of bird diets, again, for the same reason, and so on. So the potential exposure of animals to these mosquitoes is likely to be quite low, but even if they do eat them, there are no effects on them.
Okay, thank you, Dr. Rose. And the flip side of this question, and this is a follow-up from Christina, who asked one of the earlier ones. She's saying, if the mosquitoes are eliminated, why doesn't it impact other animals? And anyone can respond to that. I just want to make sure that we're being responsive to those questions about other animals.
I think we've answered the food source piece, which is, if we eliminate them and take them away as a potential food source, other animals, other predators eat other things. So they'll go back, and continue to feed on the native species that are in plentiful supply. So in terms of taking it out of the food chain as a food source for predators, it is essentially benign. So I would say that's that aspect covered.
Thank you very much, Dr. Gorman. Another question that we've gotten from Deborah Wilson in Marathon, and I know this came up in an FKMCD board meeting. She says, since this is a new product from the previous vote 4 years ago, can the present residents who live here now have a new vote on this new product? I don't know if FKMCD may want to respond to that.
Sure. So I know that it has been discussed at previous board meetings, but as of now, it is not on any agenda. So I would just encourage you to reach out to your commissioners if that's something that you're interested in. But at this point in time, it's not in any sort of plan that we may have.
Thank you. Believe it or not, I think that we may be running out of questions. I'm trying to not be repetitious. We have received a number of submissions from Barry Ray. They're not all questions. If you all could just give me 1 second, make sure we're not missing anything. And I think some of the panelists have access to this Q&A box. Just want to make sure we're not missing anything. Give everyone an opportunity to ask their questions, especially from locals. Okay, well, here's one. Sorry, I'm getting help with these. Okay, here's a question. This was from Kim Sikora, Key Largo resident, and she's asking, why did Oxitec target the Florida Keys? Why not Marco Island, St. Petersburg, Satellite Beach, Daytona, Panama City, Naples, or any other coastal town? And it's a question for Oxitec. So Dr. Gorman, would you like to respond to that? It's, why did Oxitec target the Florida Keys? I can answer part of it because I know we were excited, and why not these other places? But Dr. Gorman, maybe you'd like to speak about that.
Sure. The initial contact was through discussion with, I think, Andrea Leal's predecessor, and Oxitec. The technology had been heard about, and an inquiry was made. It's an area of the US where disease risk is probably a large threat. Disease from Aedes aegypti, of course. Each year, Florida suffers at least some disease cases, and some years are much worse than others. In terms of our mosquito, it can work anywhere where the wild mosquito is. So it really doesn't matter whether it's a coastal area or whether it's, you know, a inner city area in a large metropolitan. It can work anywhere where the wild mosquito can survive. So can ours too, of course. So there's no restriction in that sense. I think it was really, you know, the forming of a strong partnership because of the need here and the proactive nature of the Mosquito Control District, who are looking for new tools and want to do as good a job as possible. Andrea?
Yeah, I mean, this really started when we saw a presentation given about the work that was originally happening there in Cayman in 2009 and 10. So it wasn't the director before me, but the one even before him, Director Ed Fussell, originally had started this process. So again, 9 and 10 were big years for our district. It was the first time we saw locally acquired mosquito-borne diseases in decades. So we really were looking at every possible technology and seeing the initial information coming out of Cayman. Really, we just wanted to follow this and see if there was something that we could potentially be doing here in the United States as well.
And Meredith, if I might continue with that, to get back to the 9-10 period of time, which I was here then too, is there was, like I say, this was the first sustained outbreak of dengue fever in the United States, especially in Florida, for years and years and years. And the health department working very closely with mosquito control, looking at how you could contain the outbreak of the disease, is what were the available options. And there weren't a hell of a lot of available options at that point in time, to be perfectly frank, unless you were gonna saturate the keys in some sort of insecticide, but that didn't really work either because of this mosquito. So, I mean, the benefit of this from a public health standpoint is you reduce the chemical imprint that you are putting into the environment. Plus, also from my perspective, when it works and looks at it, is you have a way that naturally the mosquitoes involved, either the wild or the Oxitec mosquitoes or anyone like that, actually go after one another. In other words, it's not like the human species trying to figure out where this mosquito is hiding. It is the natural reproductive cycles. They seek one another out, and that was beneficial. Also, there was an incredible amount of interest from other health departments. I remember Harris County, Texas, and also I was on a panel with them, and a national panel with them, and also from California, as far as how do we go in public health? What's the next step going to be? So this is what was really found, and a lot of the other innovative technologies that we've seen have not panned out in the decades since this was started. So why the Florida Keys is because we had an actual outbreak of a mosquito-borne disease here. Other places in Florida didn't, but I do know that we fielded lots of questions at that time and still do when we have recurring outbreaks of locally acquired dengue. And that's happened periodically since 2009, 2010.
Thank you, Mr. Eady. Any other responses from the other panelists? Okay, We did get a number of additional questions, including from locals. I did want to address just with Barry Ray. We have received the submissions. We're trying to pick through the questions. We think we've covered female releases in our first generation mosquito. We've talked about community engagement, the questions on opt-out, risk assessments, Cayman, tetracycline, and the referendum. So we think we've touched on all that, but it was covered by a few panelists across a few answers. And one question here, this is from Megan Hull, and then we have a couple others from locals. So are we willing to debate other scientists who are concerned? That's the start of her question. I think there's a little bit more, but Dr. Rose, could you talk about our interactions just with other scientists and independent and peer review? This is for Oxitec.
Yeah, so thanks for that question as well. So with regard to the regulatory process, the EPA is mandated by Congress to assess these kind of applications scientifically, and the EPA use probably a dozen scientists or more who are experts in various fields, molecular biology, entomology, public health, ecology, and so on, when they actually assess this application. So every aspect of this technology has been picked over in great detail by the scientists at the EPA. And that was an exhaustive process, a long process, and required a huge amount of data from Oxitec. After EPA issued their approval, the Florida state authorities then also assessed this application, and 7 different agencies and departments within Florida assessed this as well. So we had people at the Florida Department of Health, people at the Florida Department of Agriculture and Consumer Services, and various other state agencies, including the Florida Fish and Wildlife Conservation Commission looking at this as well. In addition, we had CDC scientists who were commissioned by EPA to assist with certain aspects of the review that were related to human health and the questions about hybrid vigor and introgression that we dealt with earlier. So if I count all of those people, we're looking at dozens of independent scientists who have actually assessed this technology in great detail. In addition to that, Oxitec, as I said earlier, has published well over 100 peer-reviewed papers on this technology in a variety of its insects, including mosquitoes. So we've had significant interaction with scientists when they actually come to assess this technology.
Thank you very much, Dr. Rose. Would anyone like to add anything else? Okay. Dr. Gorman, I will send a question to you. This is more from Barry Ray.
Okay.
He says we have not addressed Cayman data. So if we could talk about the Cayman question around performance numbers. We've talked about that a little bit, but could you share that again, please?
I certainly can. Yeah, happy to do so. So without wanting to go, forgive me for going over old ground, and I know it's been a fairly long session, so I will do a little bit. 4 projects in Cayman, all first generation mosquitoes, ranging from 2009 to 2018. 2009 being the first one and the first ever GM mosquito release, so a pioneering project, and the achievements there were really around the biology and looking at the feasibility of this kind of technology. And it was the following year, the 2010, where an 80 to 85% ovitrap index reduction was documented over, I think, a 7-week period. That was a 7-week assessment period for that. So that was when the performance measurements started, if you like, for 513A. 2 further projects since then, 2016 and 2018. Different metrics mean that you get different numbers. So I would say that the overtrap index is one that's sometimes used. Eggs per trap is another one, typically more sensitive and more accurate. Overtrap index is a bit like saying how many trees, did you have in your trees of fruit did you have in your yield, rather than how many fruit themselves. So actually counting the eggs as opposed to counting the traps that had eggs in them tends to be more accurate. And so there have been various reanalyzing of the data. That 80 to 85% overtrap index in the second project translate on a 4-week eggs per trap of 96%. So it just depends on how you slice that data as to which metric you feel is the most appropriate one, depending on the circumstance and the projects. The 62%, which was achieved in eggs per trap, I think that again was a 7-week average, the 62% on a particular period. That was the lowest, certainly lower than some of our other pilot studies. But again, as I mentioned before, it was on the back of a brand new rearing facility, which was a whole new model that was being tested at the time. It was essentially a single season. And you know, it was a pretty strong result considering some of the background complications. 62%, not too bad, but when you compare it with other technologies, particularly conventional ones. And then there's the 2018 project. Again, that was assessing suppression as well as a range of other things, but alongside other technologies. So here it was in rotation with chemicals, so larvicides. It was also, this project was also using reduction of breeding sites, essentially physically removing breeding sites and spraying of breeding sites on the ground, as well as aerial BTI sprays and Oxitec, all combined in a particular strategy. The results of that project are still being analysed, and you know, agreement of the steering committee will unfold and turn that into a publication at some point soon. But those particular percentages we still can't talk about because of a mutually agreed confidentiality clause in the contract, which just meant that the steering committee had to approve any publication of results, and that hasn't happened. So I'm not at liberty to talk about actual percentage numbers for that 2018 project, but hopefully I've covered the other ones. Meredith, you're on mute.
I'm sorry. Thank you, Dr. Gorman. If anyone else has anything to add, you're welcome to. Okay, we'll move on to the next question. We do have a few more from local residents. This is from Katie in Key Largo. This is a 2-part question. The first part will probably go to Dr. Rose. Says, will Oxitec publish or make public the thousands of pages submitted to EPA for the public to view? So we'll let Dr. Rose answer that. The other question says, in regards to the thousands of dollars that Oxitec donated to the Mosquito Control Board, will they be required to reimburse Oxitec should the experiment be rejected? I'm not aware of any Oxitec donations to the Mosquito Control Board, but I guess someone can tackle that question after we've gone through the first one, which is, will Oxitec publish or make public the thousands of pages submitted to EPA for the public to view? And Dr. Rose, if you could start with the first one.
Yeah, thank you. So let me just break down what those thousands of pages consisted of, because that'll help to explain the answer. Part of that is data relating to the actual field trial protocols and the methods that we plan to use to actually assess how these mosquitoes work. That's already been published. Part of the submission is all of the data related to the risk assessment. So that is things like the impact on human health, on animal health, and so on, which the EPA used to make their risk assessment of no risk to humans or the environment. So most of that data has already been summarized by the EPA in the human health and environmental risk assessment, and that is already publicly available as well. Beyond that, there is also data which is submitted to EPA, which has to do with production methods, quality control methods, and various things like that, which are confidential business information. And obviously that is not something which Oxitec can make public. So much of that information in those thousands of pages is already publicly available.
Thank you, Dr. Rose. I mean, the second question, I'm not sure what to do with that, because we did not donate money to the Mosquito Control Board. Does anyone from Oxitec want to say anything additional, or from FKMCD about that?
Andrea, maybe?
No, I mean, we've not received any funds whatsoever from Oxitec to the district. So again, as far as individual commissioners go, I know that they've adamantly denied any such accusations as well in public meetings. So I don't honestly know where the information is coming from.
Thank you.
Yeah, and I just add that it's it's an Oxitec funded trial, and therefore all costs are being covered by the company.
Okay, thank you. This next one is from Kim. I believe her last name is pronounced Sikora, in Key Largo. She says, if I get bitten by a male mosquito treated by tetracycline and scratch it resulting in an infection, specifically a staph infection, will tetracycline, doxycycline work to eradicate my infection? Dr. Rose, would you, I mean, we've talked about tetracycline, but I mean, perhaps.
Yeah, I can pick that up.
Thank you.
Yeah, so the first important point to make is that male mosquitoes don't bite anyone. So there is no possibility for any resident to be bitten by a male mosquito. The second thing is that the male mosquitoes which are released are not treated with tetracycline. As I said earlier, tetracycline is used only in the United Kingdom in small quantities to help keep females alive to produce eggs. But the male mosquitoes released in Florida have never been in contact with tetracycline. So therefore, the rest of that question doesn't apply.
Okay. Thank you, Dr. Rose. The next question, this is also from Deborah Wilson, and I mean, we've talked about this a lot, but we'll just address this one directly. It says, even though it is not required of you by regulators, why do you not perform a simple and inexpensive test to check for the presence of antibiotics on your mosquitoes once they are released? Dr. Rose, do you mind taking that one too?
Yeah, that's fine. So as I've said this evening already, regulators have assessed this and determined that there is no risk of the spread of antibiotic-resistant bacteria in the Keys. So again, this does not apply. We don't test anything in response to members of the public. We test only in response to data required of us by regulators. And any company would have the same position.
Thank you, Dr. Rose. We're running low on questions, so we are going to answer a question. This is from Dana Pearls. She's not from the Florida Keys. She's with Friends of the Earth. And she says, given that COVID is on the rise in Florida and Oxitec technicians will need to access people's yards, how will Oxitec be able to collect data, traps, boxes, etc., and be in people's yards without putting people at risk of COVID exposure? I'll hand this to Dr. Gorman, who is Oxitec's head of field operations, and unless others would also like to weigh in, and in which case you're welcome to.
Well, I can start, Meredith, certainly. It's a great question, and it's something that we'll be working very closely with FKMCD to resolve as well. It's unprecedented times and very unfortunate. We've had the experience of working in Brazil under COVID, and we've had field projects which are ongoing, and they were releasing mosquitoes as well as putting out traps and bringing back those trap catches. So we've managed to continue operations right the way through, to a certain level. We've had to amend our operations. We've had to make sure that we're operating outside houses, for example, rather than inside closed areas. We've had operational changes to make sure that we can continue to do those things. But in particular in Brazil, dengue is a real threat still as well there too. I mean, a really solid threat. And the public health officials are really worried about mosquito-borne diseases just as they are about COVID-19. So there's always a play there between the two, and the risk versus the reward, if you like. But when it comes to the operations, we'll be able to run operations sensitively, but we are extremely aware that the COVID-19 situation in the US is continuing to be a problem, and in Florida it continues to rise. And so our timing with FKMCD will take all that into account, and we'll be extremely sensitive. The safety of our staff, safety of the residents is absolutely paramount and takes priority over anything else.
May I say something here?
Yeah,
yes,
please.
With regard to COVID, and Oxitec, like anyone else, would be required, that we're saying that you need to wear face coverings, but you have to be within 6 feet of someone really to have a legitimate chance of catching COVID. If you have a mask, that chance of going down or the chance of catching the disease is very, very small. So, I mean, the message that for this call is just like the message we have had in public health from the beginning, except with the addition of wear a mask, keep social distancing. So in this case, if someone is coming in to pick up a box or something like that in the outside, make sure as an individual you stay away from that person, and wear your mask, wash your hands when you go back in. So, I mean, I am assuming that from what I just heard, those are protocols that the Oxitec, and I know that mosquito control personnel are already following those, as are the other utilities in the Keys. So, it's up to individuals now to make sure that they're wearing their mask, that they're socially distancing in every chance that they get, washing their hands. If they're sick, stay home. And I'm saying that just as a public health message, not just in this context, but in any context. So the thing is, individuals can control more than anything a government or anybody else does, their exposure to COVID-19. But individuals have to take their responsibility very seriously and do what they've been asked to do.
thank you. And Ms. Leal, is there anything that you might want to add here? I mean, I know your team is having to operate within these constraints today.
We are, and we are operating at full capacity, which means, we are going on people's properties. We're going door to door. We're still educating homeowners, dumping standing water, treating standing water, placing traps, whether it be adult traps or overcuts on private properties. But again, as Bob Eady said, we're doing so in a responsible manner. We're making sure that all of our employees have masks, gloves, whatever they need to make sure that they're protected and residents are protected. We are practicing social distancing and basically following all of the guidelines that our health department is giving us to follow. So I think it's important, whether it's everyday operations or a trial, or, at home, as Bob's saying, you're going out for a walk at the end of the day, making sure that we're following those guidelines. And they are required to be followed by our district. And I'm sure, should we collaborate with Oxitec or any other company, they will be required to as well.
Thank you very much. We have 2 more questions. If no others come through, just 2 more. One is from Melissa Stone. She asks, will this be able to be adapted to be used in other regions with different species that carry other diseases? Perhaps in places, she says, where there's greater need, maybe with malaria. Certainly Oxitec can respond to this. If any of the other panelists are interested in speaking on this point, you are welcome to. Okay, I'll hand it over to either Dr. Gorman or Dr. Rose. I'm not sure which one of you might want to.
I can take that one, Meredith. I think it's a great question, and one that is very pertinent, because we've quite recently, or relatively recently, started on strains to combat malaria too. So different species, different geographical regions have different disease threats, but mosquitoes are present just about everywhere. So it's a species-specific technology. That's the one thing to remember. So it only works against the species that it's targeting. It doesn't work against any other species. So it means that you have to, you basically have to put the technology into another strain of mosquitoes. So it's not a quick process, but it is one that we can do, and we're pretty good at transforming mosquitoes now. So the Gates Foundation made a multi-million-dollar investment to help for us to fund some malaria strains, 2 strains of Anopheles for use in the Horn of Africa and potentially in Central America, should they get that far. And the projects are going extremely well. We've had lots of success. We also have Aedes albopictus, which has already been transformed and essentially waiting on the shelf. So with the right pull from people in need, we can put this kind of technology into into most mosquitoes. They're all different, but we can certainly get it into most. And hopefully have a real impact on not just with Aedes, dengue, Zika, yellow fever, but also with Anopheles and malaria too.
Thank you very much, Dr. Gorman. Any others wishing to speak here? Okay, we have gotten more questions, and one from just a few minutes ago asked, this is Drew, asked, so this will reduce numbers of mosquitoes to reduce pathogen prevalence without causing extinctions, is the question. And Dr. Gorman, I don't know if you might want to respond to that one too.
I can, but I mean, I'm happy to hand it over to anybody else if anybody else wants to tackle it too. No? Okay, well, certainly the link between disease transmission threshold and mosquito prevalence is one that's not a straight line. So in other words, you can't just pull down your mosquito abundance and expect an absolute corresponding reduction in disease transmission either. There are a whole load of influencing factors there. And so large epidemiological trials, WHO, World Health Organization style, that cost tens of millions of dollars and are done over multiple years to demonstrate health impact, are great and may happen in due course. And are primarily targeted towards malaria in that sense. But it's certainly the case that without the vector, you don't get disease transmission. So there is a reduction in disease transmission when you reduce the vector, but the actual relationship is very variable and depends on lots of other circumstances. So we would expect a high reduction in the pest to result in a substantial reduction in disease. But as I say, each situation does differ a little bit. Depends on the disease, depends on the vector, but also the local conditions.
Thank you, Dr. Gorman. For the next question, I'll turn to Dr. Rose. The question is, when will the final product be available after all of your proposed trials? This is really more of a regulatory question, I believe. Dr. Rose, are you
thanks for that
still with us?
question.
Okay.
Yes. So the process, once this particular project is completed, is obviously to analyze the data and then to submit an application to the EPA for product registration. That then has to be assessed by the EPA, and the exact timeline for that can be variable dependent on exactly how long they take to do their data analysis. So I wouldn't be able to give an exact timeline for that. But that is definitely the next step and the most important regulatory step before this would be a product which could be used more widely.
Okay. Thank you, Dr. Rose. Any comments from anyone else?
Meredith?
Yes.
Meredith, this is Larry Barr. Could I go back to a previous question somebody asked? I should have spoken up earlier, but the response has been kind of percolating in the back of my mind. Was I correct? Somebody asked what happened if she would be bitten by a male mosquito.
Well, I think they might have said an oxy. Yeah, but you're right. I mean, they can't be bitten by an Oxitec mosquito.
Well, I thought I stated if bitten by a male mosquito.
You know what? You're right. That yes, that's not possible.
Okay. Well, let me. Yeah. You know, mosquitoes and other blood-feeding flies, they're not like other animals or even many other insects. For example, you look at you take a male and a female dog; their mouths are the same. Look at human beings, yours and mine. If we stood together, our mouths are the same. These blood-feeding flies, the morphology is actually different between the male and the female, where the females have mouth parts that are adapted for piercing skin and obtaining a blood meal, whereas the males do not. I mean, you can, if you have the patience and a good enough microscope, you can actually dissect out the various parts that form the mouth parts of a male mosquito and a female mosquito, and they're different. And the male mosquito simply does not have the structure that would allow it to pierce skin. Thank you very much.
Thank you, Dr. Rebar. Okay, I think there might just be one more question, one or two, and we'll answer these quickly and then give the panelists an opportunity just to say something closing before we get cut off, I think in about 13 minutes. Most of this has been discussed, but this is from Deborah Wilson. Since chemical spraying will continue during the experiment, how will the causal factor of population suppression be determined? How will you know how successful your trial was? And this could go to FKMCD or Oxitec. Dr. Gorman, I don't know if you want to take it or...
Andrew's going to go first, I can tell.
Yeah, I'm going to jump in first. So basically, one of the things that we wanted to ensure when doing any trial is to make sure that it fits into what we're already doing. So whatever we're doing in these trial areas, we're going to be doing in the comparison control areas. So that way we can make sure that we're not necessarily impacting the study, but we're going to really get a good idea of suppression in areas where we're doing everything that we possibly can.
Okay, thank you. And we do have two more questions, if we can squeeze these in. These are from, we just received a lot of questions from Diana Reeves, GMO Free USA. And we're going to focus on two of them. One says, have you taken into consideration the Tovey Lehman peer-reviewed paper about windborne mosquitoes that was published in October 2019? This is hurricane season. Windy weather is likely, which raises containment issues. There is no telling where the GM mosquitoes could end up. Dr. Gorman, would you like to take that one? Okay.
Yeah, and Nathan, feel free to fill in if need be. But Yep, these kind of studies fit into two large groups, really. There's contained trials and there's open release trials, and this is an open release trial. It's quite conceivable that mosquitoes may travel. They do travel, and the EPA have considered that, as have all the other regulatory agencies who've been involved in the decision. So there are boundaries around where we can release, but we can't put boundaries around where the mosquitoes go. What we can do is we can monitor and we can work out how far they travel and how far that they go. But there are some unknowns there, and we will, if the weather gets bad, we do have a threshold there where we will bring in the release devices, we will bring in the traps, operational thresholds for bad weather, etc. Bad weather where we wouldn't release, only certain conditions where we'd actually do the releases, etc. But you can't take away from the fact that this is an open release trial, and mosquitoes do travel, and be that jumping into somebody's car or being blown on the wind. Those are circumstances that have been taken into account in making the regulatory decisions.
Thank you very much.
Yeah.
Anything else from anyone else?
Could I just add something there?
Yeah.
Yes.
This is Nathan Rose. Just to say that the mosquitoes are not going to persist in the environment, whether they are in the release site or whether they were to be outside the release site. These mosquitoes disappear from the environment within a few generations. So again, when it comes to assessing any potential risk with mosquitoes moving, this is something the EPA has taken into account, and these mosquitoes will disappear from the environment quickly.
Thank you very much. This is going to be our last question. It's from the same person. It's from Diana Reeves with GMO Free USA, and it says new research by Tang and Davis published in Communications Biology indicates that climate change could enable mosquitoes to mutate more rapidly. This could have a profound effect on any release of experimental mosquitoes. Have you considered the impact of this research? Dr. Gorman, I don't know if this might be one for you or perhaps for others.
Yeah, I think it's a more general question. I don't know whether one of the other panelists wants to take a go at this one. It's the last question.
Yeah, no, in fact, I don't know, Dr. Ray, not to put you on the spot, but this may be a good one for you or really anyone, but it's new research by Tang and Davis published in Communications Biology indicates that climate change could enable mosquitoes to mutate more rapidly. This could have a profound effect on any release of experimental mosquitoes. Have you considered the impact of this research?
Okay, I'll start. First of all, I haven't read that paper. I don't even know what journal. I don't recognize that journal. I think what they may be referring to is that as many insects, the warmer the climate, the faster the generations go through, and then the faster the generations, the faster that mutations can take place. That's, I think, what they're referring to. The other side of an argument like that is that climate change also projects that some areas will get warmer, some areas will get colder. So it's really difficult to tell what the net effect is going to be. It may be faster in one area and slower in another. I think that's what they're referring to. And while I got the floor, I'd like to, this could be taken as my closing remarks too, is that it needs to be understood this is a field experiment. This is not an experiment in the lab where everything is controlled. So wind and changes in weather, that's part of field research. I've been in field research all my life, and we struggle with that. But this is a field experiment, and if you're going to be realistic about what the results are going to be in nature, you're going to have to deal with all of these, which I think Oxitec is doing the best they can to deal with those things.
Thank you, Dr. Ray. And thank you all for your time and attention tonight. I mean, the panelists and everyone in the audience, it's past dinner time. We've been here for hours. And thank you. But I do want to give a final opportunity for any of the panelists. If you'd like to make any final comments, you're welcome to now. We just have about 6 minutes left.
Meredith, I'll go first. I'd like to think, that that everybody else will say something afterwards. But just a great big thank you from me to all the audience for tuning in, if you like, and taking part. That was a really good barrage of questions we got through. Well, I'll find out afterwards how many questions we got through, but certainly a very good number. So thanks to everybody who popped in a question, but also thanks to those who just tuned in to listen. Thanks to you and Ben, particularly for moderating and fielding all those questions so quick. And yeah, special thanks to all the panelists, all colleagues on the panel here, because yeah, I thought it was a great session. So I'm much obliged to everybody.
We appreciate it, and Dr. Ray and Mr. Eady and Dr. Mader are all part of our independent advisory board if this project moves forward. So we'll continue to work with them, and we appreciate the time and attention of everyone.
Thank you.
Okay. If nothing else, we'll say goodbye and good night, and just thank you. No, and I mean, especially to FKMCD and to Ms. Leal and Dr. Rebar. I mean, this project has taken a lot of your time over the last decade, the idea of this project, and we appreciate your patience, and we thank everyone for their interest.
Yeah, and I think before we leave, I want to make sure that everybody that's still hanging around, there are a few questions that kind of lingered. So we'll make sure that those questions are answered. Any further questions that anyone may have, you can contact Oxitec, or you can contact the district at questions@keysmosquito.org. We want to make sure that everyone's questions are answered and everyone has a chance to get the information that they're seeking. So please, we ask that you continue to reach out with any further information that you may need.
Absolutely, and Oxitec is responding to questions too at florida@oxitec.com. And I hope we covered everything this evening, but if we didn't, or if additional questions come up, we welcome them. And I think Mr. Eady might be wanting to say something, or he might have just been waving goodbye. He might have just been waving goodbye.
I think the latter, yeah. Yeah, I think the latter.
Okay. Thanks, everybody.
Thanks so much. Really appreciate it. Thank you.
Good night.
Good night.
Good night.
Good night.